Hello and welcome everybody to my channel ominibioentrance, and today we will be seeing CSIR PYQs June 2023 Shift 2. I will be dealing with applied biology and methodology, DNA replication, protein synthesis and genetics. These are solved questions of part C. If you are new to my channel, please do like, share and subscribe. I have also uploaded old CSIR PYQs. You can refer to my channel for the same. Next question. The following statements were made about laser scanning confocal microscopy LSCM. LSCM is wide field technique with color illumination system. Now confocal imaging, it does not use wide field imaging. What happens in wide field imaging? What is happening there in wide field imaging? In wide field microscope, the entire specimen is flooded evenly with the light from the light source. All part of the sample is excited at the same time and the resulting fluorescence is detected by a photo detector. This is what is happening in wide field microscope, but this is not the principle behind laser scanning confocal microscopy. Confocal microscope, they use point illumination and a pinhole placed in front of the detector eliminate out of focus signals. And the image's optical resolution is much better than that in wide field microscopes. So statement A is not correct. We will see statement B. Spatial resolution is higher than that achieved in wide field imaging could be obtained if only central portion of the airy disk is used to form an image. That is correct. Scanning mirrors sweep the excitation beam over the sample point by point to build the image. That is also correct. I just explained these just now. Then, an altered pinhole size does not make any impact on the resolution of the image. It makes an impact, resolution is decreased. So D is not correct. The photomultiplier tube in LSCM helps in generating real color of fluorophores. That is also not correct. So B and C are the correct answers. So the answer here is option 3. Next question. Following statements are made about the bacterial ribosomes and their functions. You have to find out which is the combination of both correct statements you have to find out. So first statement. Association of 23S rRNA with 16S rRNA is essential to catalyze in vitro peptide formation during using model substrates. That's not correct. The next one, the 23S rRNA is necessary and sufficient to catalyze in vitro peptide bond formation using model substrates. That is correct. Ribosome carries a polymerization activity. That is also correct. The 16S rRNA is necessary and sufficient to catalyze in vitro peptide bond formation using model substrate. That is not correct. C. Ribosome carries polymerization activity. It is involved in protein formation. So it carries polymerization activity. So B and C are correct. Answer is option 2, B and C. Then shown below are CD spectra of a protein recorded under two different conditions. From the options given below, select the one that is the best interpretation. Now if you see the diagram, this is what the CD spectra of an alpha helix. This is CD spectra of an alpha helix. This is that of a beta sheet and this is that of a random coil. So here if you see in the diagram, A is that of alpha helix but B is that of a random coil. That means A is been converted to B. That is A alpha helix has been converted to a random coil. Now we will read the statement. The protein A has alpha helical secondary structure under condition A that is denatured under condition B. That is correct. That's what I told you just now. That A which was having an alpha helix got converted to B which is a random coil. So that itself is the correct answer. The protein has alpha helical secondary structure under condition A that is converted to beta sheets, not beta sheets to random coils. The spectra represent the tertiary fold of a protein. No, the difference between spectra under condition A and B is due to lower protein concentration. No. So only one is the correct answer. Then next one. A founder population has capital A small a heterozygous genotype with frequency 1 and no individual is with either capital A or small a genotypes. With repeated self-fertilization, the frequency of capital A small a after 3 generation will be. So they have asked for the third generation. So initially 100% heterozygous. So in the first generation, heterozygous will be divided to form 50% heterozygous condition and 50% homozygous. Of this 50% homozygous, 25% each will go to capital A and small a. So first will be half, capital A, small a will be half, that is 50%. Then small a will be 1/4 each. In the second generation, it will be 1/4. Out of 1/4, the remaining 3/4 will go to the homozygous form. That is, half of the 3/4 to capital A and half of 3/4 to small a. That is, capital A will be minus 3/8 and small a will be 3/8. Not minus, I am sorry. Capital A will be 3/8. Similarly, if you do for the third generation, you will get 1/8, 7/16, 7/16. So the answer here is option 2, 7/16, capital A, 1/8 will be capital A, small a, and 7/16 will be small a.